Okay, so welcome to this video. In this video what we're going to discuss is G-protein coupled receptors. Okay, so let me outline the structure of this video for you. What we're going to start off with is a discussion of the basic structure that all G-protein coupled receptors share. Okay, then what we will do is we will discuss the 5 different families of G-protein coupled receptors. Okay, then what we will do is we will move on to discussing the structural diversity of G-protein coupled receptors. Okay, so we'll look specifically at how the transmembrane regions of G-protein coupled receptors can vary between different examples of G-protein coupled receptors. Then what we will move on to is discussing what happens when a ligand that is an agonist binds. Okay, so what conformational changes occur to the G-protein coupled receptor that constitute activation. Then what we will discuss is the heterotrimeric G-proteins. Okay, so we will look at firstly the diversity of heterotrimeric G-proteins. So we'll look at all the different alpha subunits, all the different beta subunits, and all the different gamma subunits. And we'll then look at the structure of heterotrimeric G-proteins in quite a bit of detail. Indeed, we will look at the actual structure of the alpha subunit of heterotrimeric G-proteins, and we will do that so that we can then get a decent understanding of what actual conformational changes occur in the alpha subunit when it binds to the activated G-protein coupled receptor that allow the release of GDP. Okay, then what we will do is, after discussing the binding of the heterotrimeric G-protein to the G-protein coupled receptor, we will discuss the binding of GTP to the heterotrimeric G-protein and how that then leads to the heterotrimeric G-protein falling apart, basically, how it leads to the alpha and the beta-gamma complexes dissociating. We will then briefly talk about the signaling that the alpha subunits and the beta-gamma subunits participate in. And then what we will talk about is how you turn the alpha subunits off. Okay, how you get them to hydrolyze the GTP. So we'll talk about regulators of G-protein signaling, okay, or RGS proteins. Okay, then what we will talk about briefly is the dimerization of G-protein coupled receptors, and really the main example there is the GABA-B receptors, so we'll look at those. Then what we will move on to is a discussion of the desensitization of G-protein coupled receptors, so how you can turn them off using the G-protein receptor kinases and then the arrestins. Okay, we'll then talk about arrestin signaling. Okay, so how arrestin molecules can activate all sorts of cascades that you don't generally think of as being associated with G-protein coupled receptors. And then finally we'll talk about biased agonism, which is this concept that different ligands can alter the conformation of G-protein coupled receptors in different ways, and that these different conformations of the G-protein coupled receptor, which are all active but are different, can then determine the specificity of what that G-protein coupled receptor is actually going to activate, so the specific downstream pathways that the G-protein coupled receptor is going to activate. Okay, so that's where we'll end up with this. Okay, right, so let's start off with the basics then. Okay, so the first thing that I want to say is that there is a movement within pharmacology to have G-protein coupled receptors renamed. Okay, and the new name that people want to use for G-protein coupled receptors is to call them 7 transmembrane receptors, which for short would be abbreviated down to 7 TMRs, like so. Okay, so this stands for 7, and then the TM is for transmembrane, okay, and then the R is for receptors. Okay, so 7 transmembrane receptors. Right, so why do people want to have them renamed that? Well, the first thing to say is that it's a sensible name because they do all have 7 transmembrane alpha helices, as we'll see in just a moment. Okay, but what's wrong with G-protein coupled receptor? The reason people want to turn away from G protein coupled receptor and over to this 7 transmembrane receptor name is that, as I just said, the arrestin molecule, not necessarily just beta-arrestin, there are arrestins other than beta-arrestin. The arrestin molecules, which can bind to G protein coupled receptors as part of the desensitization pathway, can actually activate all sorts of different signaling cascades, okay, that do not involve heterotrimeric G proteins. For instance, they activate cascades that you would think more usually as being associated with receptor tyrosine kinases rather than G protein coupled receptors. Okay, and that's why people want to move away from the name G protein coupled receptors, because it implies that all these receptors do is activate G proteins, when in fact they have a huge number of other signaling pathways that they are also capable of activating. Okay, so that's the clever reason why people want to move over to using this name of 7 transmembrane receptors rather than G protein coupled receptors. Okay, because of the fact that actually these things do have actions outside of heterotrimeric G proteins. Okay, right. So, next thing to say is that you have an absolutely huge number of different G protein coupled receptors in humans alone. Okay, so in humans alone, we have greater than 800 different G protein coupled receptors. Okay, now some of these we don't actually know what they do, but they're in the minority, the ones that we don't actually know what they do. Okay, they are known as orphan receptors. So how do we even find these orphan receptors if we don't have a clue what they do or what their ligands are? Okay, well, orphan receptors are a consequence of molecular biology overtaking pharmacology. Okay, molecular biology and the Human Genome Project sequenced the entire genome. They found loads of new genes. Okay, they found genes that they thought were G protein coupled receptors because of how homologous they were in sequence to other genes which we know code for G protein coupled receptors, and therefore they said this makes a G protein coupled receptor. Okay, and that's where orphan receptors came from, and we're still gradually working out what the ligands for these orphan receptors actually are. Okay, right. So in humans alone, there are more than 800 different G protein coupled receptors. Okay, I think if you go to all different life forms that we have studied, the number that we have now found is over 30,000 G protein coupled receptors, and that will grow and grow and grow as we go to more and more life forms. Okay, so this really is a very, very common type of receptor, basically. Nature has found this and it has run with it, basically. And it's not totally understood why this is such a successful design. What is so good about having 7 membrane-spanning alpha helices? Regardless, we will now study the structure of G protein coupled receptors, the basic facts, and then we'll look at the 5 different families of G protein coupled receptors, which we divide these greater than 800 G protein coupled receptors in humans into. Okay, so let's start off then with the basic structural features of all G protein coupled receptors. So I'm going to draw 2 parallel lines here to represent the cell membrane. Okay, so this bottom line can represent the inner leaflet of the phospholipid bilayer facing into the cytoplasm below, and this top line can represent the outer leaflet of the phospholipid bilayer facing the extracellular fluid above. Okay, right, the first thing to say about G protein coupled receptors is that unlike many other receptors in pharmacology, they are single proteins, okay? A single polypeptide makes G protein coupled receptors, and that G protein coupled receptor on its own is functional, okay? Except for a few examples such as the GABA B receptor, which we'll come back to later, which needs to form dimers to function. Okay, contrast that to, for instance, the ligand-gated ion channels, which are also a major example of receptors in pharmacology. Those are generally multi-subunit complexes that form the ion channel, which will open in response to a ligand. Okay, so G protein coupled receptors are very simple in that they are just one polypeptide, and that can function to transmit an extracellular signal into an intracellular signal on its own. Right, so let's have a look at the general structural features of a G protein coupled receptor then. So, we will start with the N-terminal extracellularly, like so, and then this line that I'm drawing will now represent the polypeptide. It'll represent the polymer of amino acids. So they have this very famous structure of 7 membrane-spanning alpha helices, like so. Okay, now, the membrane-spanning alpha helices then, I will just colour these in now in red. Okay, now I have not actually shown them as alpha helices, I've shown them as straight lines. Now I do that because it makes the picture look nicer, it's easier on the eyes if you show it as straight lines. Okay, however, in reality, these polypeptides in these transmembrane domains here, they should be little spring-shaped structures. Okay, the polypeptide should be coiled into an alpha helix, which is a spring shape in effect. Okay, and those alpha helices which span the membrane, they'll have lots of hydrophobic residues which will allow the alpha helix to sit in the lipid core of the phospholipid bilayer thermodynamically favourably. Okay, right, so these 7 membrane-spanning alpha helices are given names. Okay, the first membrane-spanning alpha helix is known as transmembrane domain 1, or TM1. Okay, the second one is then known as TM2, and we're going from amino terminal to C-terminal. Second one is TM2, third one is TM3, fourth one is TM4, fifth one is TM5, sixth one is TM6, and then the 7th one then is TM7. Okay, right, so now let's label up some other portions besides the transmembrane domains. Okay, next let's label up the intracellular loops. Okay, so these 3 loops that we have in between transmembrane domains, this first one here between transmembrane domain 1 and 2, this second one here between transmembrane domain 3 and 4, and this third one here between transmembrane domain 5 and 6, these are known as the intracellular loops. Okay, so these are called the intra because they're inside, cellular because they're inside the cell, and then loops. Okay, and for short, the intracellular loops are abbreviated as the ICLs. Okay, so the first intracellular loop, which is the one closest to the amino terminus between TM1 and TM2, is known as the intracellular loop 1. The second one between TM3 and TM4 is known as the intracellular loop 2. The third one between TM5 and TM6 is known as the intracellular loop 3. Okay, so those are the 3 intracellular loops, and we'll talk more about those later on. Okay, let's now do the extracellular loops. The extracellular loops are the same thing, but on the outside this time, and again you have 3 of them. Okay, and for short they're abbreviated down to ECL for extracellular loop. Okay, so this first one here between TM2 and TM3, that's extracellular loop 1. This second one here between TM4 and TM5, that's extracellular loop 2. And this third one here between TM6 and TM7, that's extracellular loop 3, ECL3. Okay, right, there are 2 more important domains that I want to mark on, which are the 2 final bits that we haven't coloured in yet. Okay, so the portion that's prior to TM1, which I'll colour in blue here, this is known as the amino terminal domain, or for short it's just abbreviated to ATD. So that stands for the amino, that's the A, then T is for terminal, and then the D is for domain, so amino terminal domain. And then the domain after the after TM7, this then is the domain closest to the C-terminus. This is then called the C-terminal domain, and for short that's abbreviated down to CTD. Okay. Right, so those are the broad structural features that all G protein coupled receptors share. This is their membrane spanning topology, if you will. Okay, right, Now what I want to show you is how these 7 membrane-spanning alpha helices that are found in all G protein coupled receptors, that's the feature that characterizes all G protein coupled receptors, okay, how these form what is known as a 7-helix bundle, okay, because they don't sit all in a line like this in actual reality. What happens is they fold around into a bundle, okay, and this bundle is known as the 7-helix bundle. Okay, so let me now show you this. Okay, so basically they all fold round into a ring basically, and let me show you this. Once again, here is the membrane, okay, and then I will now draw this as a cylinder, like so. And we're going to have the 7 transmembrane alpha helices all in a ring making up the outside of this cylinder. So there's a hole down the middle where the ligand's going to bind later on. Okay, now the outer wall of this ring is going to be made up of the 7 alpha helices. So there's 1, there's 2, there's 3, there's 4, there's 5, there's 6, and there's 7. Okay, and I'll divide this up like so now. Okay, so this will be sitting in the membrane like so. So now let's try and make this look more like this. So let's say that this alpha helix here is going to be the first alpha helix, so its amino terminus, well, the amino terminal domain of the G protein coupled receptor will then be prior to that first transmembrane helix, so it will be here. So let's colour that in. So here in blue is the amino terminal domain. Okay, that then is the first transmembrane domain, which will go through the membrane, and then it will come back up, and then here is the second transmembrane domain here, and then you'll have an extracellular loop, which will be extracellular loop 1 here that we'll be able to see. So that's that first extracellular loop here, linking transmembrane domain 2 to transmembrane domain 3. Transmembrane domain 3 will go all the way down, you'll have intracellular loop 2, and then it will come up here, and now I might actually draw this. Okay, so here then is that 4th membrane spanning alpha helix, TM4 there. Okay, then we'll have the second extracellular loop here. I might just mark it in the black pen as well so you can actually see it. Okay, then you'll have the 5th membrane spanning alpha helix, then that, which intracellular loop is this now? The 3rd intracellular loop here, okay, then the 3rd extracellular loop, and the 7th membrane spanning alpha helix, and then the C-terminal domain down here. Okay, so let's colour in those different bits then. So we've got another extracellular loop here, okay, we'll colour in the transmembrane domains in red again. Okay, like so. Here's the 5th, here's the 6th, here's the 7th. Then we've got an intracellular loop there. I might try and draw a little bit of an intracellular loop here. Okay, coming from transmembrane domain 3 into transmembrane domain 4 there. Okay, and then we'll also colour in the C-terminal domain here in turquoise. Okay, right. So that's how the G protein coupled receptor actually folds up in the membrane to form this bundle of 7 membrane spanning alpha helices with that key site in the middle there. And in most G protein coupled receptors in humans, the ligand is actually going to bind in that site there. In other examples, it's a little bit more complicated, and we'll come back to that in a moment. Okay, right. So now let's actually talk about the 5 main families of G protein coupled receptors in humans. Okay, and we're going to start off with the main family, the first family, which is the one that we're going to talk most about in this video, because most of the crystal structures that we have actually got for G protein coupled receptors are for G protein coupled receptors in this first family. Okay, so this first family is called the rhodopsin family of G protein coupled receptors, named after a very famous example of a G protein coupled receptor in this family, which is the rhodopsin receptor, which is the receptor for light that cells at the back of the retina have. Okay, and that's actually a G protein coupled receptor. Now, rhodopsin was actually the G protein coupled receptor which we got the structure of first, okay? Now, that's firstly because rhodopsin is a very important receptor, and it's secondly for a practical reason, which is that it's a very stable receptor, and it proved quite easy compared to the other G protein coupled receptors to actually produce crystals of it. And the second reason it's easy to produce crystals of rhodopsin is that it's quite easy to get a very concentrated sample of rhodopsin from cow eye, basically. Cow retina. Bovine retina is the fancier way of saying it. Okay, right, so that's why rhodopsin was the first G protein coupled receptor to actually get an X-ray crystallographic structure for. Again, I think up to about 2007, 2008, that was the only G protein coupled receptor that we had the actual X-ray crystallographic structure of. Now, in the past few years, it really has exploded, and we've got loads of the things now. Again, it will continue to grow as we go on into the future. Okay, right, so that's why rhodopsin has an entire family named after it, because it was the first one that we actually got the structure of. Okay, right, so the rhodopsin family of G protein coupled receptors then. Okay, this is the biggest family of G protein coupled receptors in humans. Okay, it has 701 members. Okay, of the slightly greater than 800 G protein coupled receptors that we have in the entire human genome, 701 of them are in this rhodopsin family of G protein coupled receptors. Okay, so let me now show you the structure of a rhodopsin family G protein coupled receptor, and I will highlight the key structural features that all rhodopsin family G protein coupled receptors have. Okay, and basically, I'm going to draw you out pretty much the identical picture that I've just drawn, but I'll do it again just to highlight the points here. Okay, right, so once again, here is this 7-helix bundle made up of these 7 membrane-spanning alpha helices. 1, 2, 3, 4, 5, 6, and then 7. Okay, right. So, what are the key features then of rhodopsin family G protein coupled receptors that set them apart from other families? Okay, well, firstly you have a very short amino terminal domain. Okay, so not very big at all. Okay, so here's the amino terminal domain, which isn't very big. Okay, and then the important thing, the really, really important thing here is that the ligand binds to the rhodopsin family G protein coupled receptor in this pocket that is made by the 7 transmembrane helices, basically. So this is where the ligand is going to bind, basically. Okay, this is what's known as the ligand binding pocket. Okay, made up by the 7 membrane-spanning alpha helices surrounding it in a ring. Okay, and I'm not going to draw all of the 7 membrane-spanning alpha helices out again. I might just show them as a pink line, like so. Okay, so here we go. That speedens it up a lot. And I'll put that extracellular loop here, and then you've got the C-terminal intracellularly here. Okay, so the characteristic features then of the rhodopsin family G protein coupled receptors are that you have a very short amino terminal domain, and that the ligand binds in this ligand binding pocket, which is made up by the extracellular portions of the transmembrane domains. Okay, so it's actually going to bind to residues in those 7 membrane spanning alpha helices. Okay, and this is how most G protein coupled receptors in the human bind their ligand, because most of them are in this rhodopsin family of G protein coupled receptors. Okay, so let me give some examples of G protein coupled receptors in this family then. So I won't put rhodopsin down because it's obviously in this family. Okay, but some other examples that are notable are the adrenoceptors. Okay, the receptors for adrenaline and noradrenaline. Okay, in addition, the receptors for dopamine, D1, D2, D3, D4, and D5. Those are all rhodopsin family G protein coupled receptors, and those are evidently very important in the brain. Okay, the histamine receptors are again all rhodopsin family G protein coupled receptors. Okay, the serotonin receptors, the 5-HT receptors, are those serotonin receptors that are not cis-loop ligand-gated ion channels are all rhodopsin family G protein coupled receptors. Okay, so serotonin receptors are a weird family of receptors. You have some of them that are ligand-gated ion channels. Well, I suppose it's not that weird. Acetylcholine is the same as well. Okay, you have some of them that are cis-loop ligand-gated ion channels, which are ligand-gated ion channels, basically, that open and allow sodium ions to come into the cell when serotonin binds to them. Okay, and they're actually pentameric arrangements of 5 separate subunits making up those ligand-gated ion channels. But there aren't very many of those. Those are known as the 5-HT3 receptors, those ligand-gated ion channels. All the other serotonin receptors are G protein coupled receptors, and they are in this rhodopsin family of G protein coupled receptors. Okay, so another notable example is then the muscarinic acetylcholine receptors, which I'll just abbreviate down to MACHRs. Okay, if you're an immunologist, you might be interested to know that all the chemokine receptors are in this great family of G protein coupled receptors. Okay, and finally, my last example is the P2Y receptors, okay, which are G protein coupled receptors that bind ATP and respond when that ATP binds to them. Okay, right, so that's the rhodopsin family of G protein coupled receptors, and once again I'll stress that the key thing here is that they bind their ligand in that actual ligand binding pocket that is made up by the 7 membrane spanning alpha helices. Okay, right, so that's the first family of G protein coupled receptors in humans. Let's now go on to the second family, and compared to the massive size of the first family, all these other families are going to look rather niche, okay, rather unpopulated. Okay, so the secretin family of G protein coupled receptors then is the next family. Okay, so this actually only has 15 members within it, okay? So what then is the characteristic feature that all secretin family G protein coupled receptors have? Well, basically, again, it's how they bind their ligand. Okay, so let me show this. So, again, we'll draw out the G protein coupled receptor sitting in the phospholipid bilayer here. Okay, and again, I'll draw it as this 7 membrane spanning alpha helix bundle here. Okay, like so. So here is that central gap in between the 7 membrane spanning alpha helices that are surrounding it, like so. Okay, right, so the ligands in the case of the secretin family G protein coupled receptors are not actually going to bind within the transmembrane regions. They're going to bind on the top here, and they're actually going to bind in a sort of wedge or a little gap in between the top of the transmembrane domain portion and the amino-terminal domain. So secretin family G protein coupled receptors, the first thing I should say is that they have a much bigger N-terminal domain generally than the rhodopsin family of G protein coupled receptors. Okay, so here is this amino-terminal domain here in blue. Okay, prior to the first membrane spanning alpha helix there. And basically, the ligands for secretin family G protein coupled receptors are firstly generally peptide molecules, and they sit in that little space that we have there between the bottom of the amino-terminal domain of the G protein coupled receptor and the top of this portion that's made up of the 7 membrane spanning alpha helices. Okay, so I'll colour this in turquoise there. So that's where the ligand is going to bind, rammed in between the top of the transmembrane domain, which is made up of the 7 alpha helices, and the amino-terminal domain, which is stacked on top. Okay, and then just to add a few more bits onto here, here's the C-terminus down here. Okay, right, so that's the characteristic of secretin family G protein coupled receptors. They don't bind their ligand actually in the transmembrane domain, okay, so it hasn't slipped down and isn't binding to actual residues in the 7 membrane spanning alpha helices. Instead, it's sitting on top of them, basically, in between the top of them and the bottom of the amino-terminal domain. Okay, right, so let me give you some examples then of secretin family G protein coupled receptors. So there are some notable examples here. Okay, so the glucagon receptor, which responds obviously to the very important hormone glucagon in glucose homeostasis, this is an example of a secretin family G protein coupled receptor. In addition, the parathyroid hormone receptor is also an example of a secretin family G protein coupled receptor, which is obviously very important in controlling calcium. Okay, another receptor important in controlling calcium is the calcitonin receptor, okay, which responds to the hormone calcitonin, and again this is a secretin family G protein coupled receptor. Okay, so those are some notable examples of secretin family G protein coupled receptors in human physiology. Okay, right. Next family of G protein coupled receptors, third family, family number 3, is the glutamate family of G protein coupled receptors. Okay, right. So, what then is the characteristic feature that the glutamate family of G protein coupled receptors has? Well, actually, before I tell you that, let me tell you how many members there are in the glutamate family. Again, there are 15 members in the glutamate family. Okay, now one of them, well actually not just one of them, loads of them are the metabotropic glutamate receptors, hence why this family is named the glutamate family. Okay, so what then is the characteristic feature that the glutamate family G protein coupled receptors all share? Well, again, they have the characteristic 7 helix bundle here. Okay, now again, they have a larger amino terminal domain, and this amino terminal domain is going to have a very fancy structure in it in the case of these glutamate family G protein coupled receptors. So let me now show you this very fancy structure. So here's their amino terminal domain, and then this is how I'm going to represent this very fancy structure here. Okay, so this rather crudely shown thing here is supposed to represent what is called the venous flytrap domain of glutamate family G protein coupled receptors. So this is a venous flytrap domain. Now, why is it called that? Well, it is compared to the venous flytrap, which basically, when a fly comes into it, closes around that fly, and then the fly is digested. Okay, so basically what happens is this venous flytrap domain, these are like the two claws of the venous flytrap, effectively, the top and the bottom of it, okay? Or the jaws, maybe, is a better word to describe it than claws, okay? And when the ligand binds, it's going to bind in the middle of these two jaws, and they're going to close around the ligand, and then what's going to happen is this whole domain here that's now closed around the ligand is going to move down and bind to the transmembrane domain here, and then that's going to cause conformational changes in the transmembrane domain. Okay, so it's an example of the formation of something known as a tethered ligand. Okay, so the ligand binds up here in this venous flytrap domain, causes conformational changes all the way over there, but then you might wonder, well, how on earth is that going to cause conformational changes down at the transmembrane domain, which of course it has to do in order to transmit this extracellular signal from the extracellular side to the intracellular side, where it actually comes down here now, and the whole thing acts as a ligand which can bind to the transmembrane domain here. Okay, right, so that's the general structural features of these glutamate family G protein coupled receptors. Okay, so let me give you the notable examples then of glutamate family G protein coupled receptors. So firstly, the metabotropic glutamate receptors, okay, which I'll denote mGluRs. Okay, and there are 8 of these, so mGluR1 all the way through mGluR8, they are all in this family, okay, of glutamate family G protein coupled receptors. Okay, now let me stress that the main glutamate receptors that you have in your brain are not metabotropic glutamate receptors. Okay, glutamate is the most important fast excitatory neurotransmitter in the brain, okay, but the main receptors that you have for it are ligand-gated ion channels, the ionotropic glutamate receptors, such as the AMPA-type glutamate receptor, the NMDA-type glutamate receptor, the kainate-type glutamate receptor. Okay, those are the main ones, and they open and allow in an excitatory postsynaptic current of sodium ions generally, which then excites the postsynaptic cell. Okay, right. The metabotropic glutamate receptors are generally presynaptically located and mediate presynaptic feedback, and they are G-protein coupled receptors rather than ligand-gated ion channels, and they're within this glutamate family of G-protein coupled receptors. In addition, there's another notable example in here. We've just talked about glutamate, the most important excitatory neurotransmitter in the brain. The GABA, a type of GABA receptor, is also in this family. GABA is the most important inhibitory neurotransmitter within the brain. So glutamate, the most important excitatory neurotransmitter, GABA, the most important inhibitory neurotransmitter in the brain. Okay, and again, the main receptors for GABA that you have in the brain are the GABA A receptors, not the GABA B receptors. Okay, the GABA A receptors are ligand-gated ion channels. Again, different structure to the ionotropic glutamate receptors. The GABA A receptors are cis-loop ligand-gated ion channels, like the 5-HT3 receptors and like the nicotinic acetylcholine receptors. Okay, and they again mediate the fast transmission, okay, by opening and allowing in chloride anions into the cell and inhibiting the postsynaptic neuron. Okay, but you also have receptors that are located presynaptically for GABA, and these are G-protein coupled receptors, and these are going to be the GABA B receptors. Okay, now the GABA B receptors can also be denoted as GBRs, because this is a bit of a long abbreviation to have to write. Sometimes they're just abbreviated down to GBRs, G for GABA, B for B, and then R for receptors. Okay, right, and there are two GABA B receptors, and we'll be coming back to these later on. There's the GABA B receptor 2, there's the GABA B receptor 1, and then there's the GABA B receptor 2. Okay, right, and they are also glutamate family G-protein coupled receptors, i.e. they have this venous flytrap domain. Right, so the next family I want to mention is the family that generally no one ever knows anything about, which is the adhesion family of G-protein coupled receptors. Okay, now this is a rather strange family. This contains a bunch of G-protein coupled receptors which bind to components of the extracellular matrix. Okay, so it contains 24 different G-protein coupled receptors. Okay, and the characteristic structural feature that they all share is they have massive, great amino terminal domains which are involved in binding to the components of the extracellular matrix. So let me just show this. So here are, again, the 7 membrane spanning alpha helices forming the 7 helix bundle here, like so. And now I will show the large amino terminal domain coming up here. So a very, very large amino terminal domain here. Okay, and this is then going to be binding to components of the extracellular matrix, which I'll demonstrate here. So this, in red here, this can represent a component of the extracellular matrix. Okay, so this is the extracellular matrix. And this is the extremely large amino terminal domain here in blue. Okay, now, unfortunately I don't know anything about these either, so I can't give you examples of adhesion family G-protein coupled receptors, but that, nevertheless, is the general structural features that these adhesion family G-protein coupled receptors do have. Okay, right, so to move on to the 5th and final family of G-protein coupled receptors, which is the frizzled taste 2 family. Okay, oh, and I should say, I don't know if I've stressed this enough at any point. How did people decide how to put G-protein coupled receptors into families like this? Well, it wasn't actually on the features that I have just described to you. These are the features that generally they all have. Okay, but the actual way that we did this, putting them all into these 5 families like this was actually done on the basis of sequence identity. Okay, so we went through all the different amino acid sequences of the different G protein coupled receptors and said which ones are very, very similar, which ones have a large enough sequence identity that they probably have a very similar structure, and that's how we group them together, okay, which is how these Frizzles and TACE2 receptors got grouped together when they seem to do very little in common with one another, but they are quite common when you actually look at their primary structures. Okay, right, so Frizzled and TACE2. So this family is named after its 2 major members, which are the Frizzled receptors, of which you have many, and also the TACE2 receptors, which again you have many of. Okay, now, the features that they all share is, again, they have to bind their ligands to an extracellular amino terminal domain, okay? And then the extracellular amino terminal domain has to then somehow translate the change in conformation down into the transmembrane domain. Okay, so once again, here is the 7 membrane spanning alpha helix bundle here. 1, 2, 3, 4, 5, 6, 7. Okay. And again, they'll have a large amino terminal domain here, or a reasonably large anyway. So here's their amino terminal domain, and then they'll bind that little ligand here. Okay, right. So the 2 major members then of this family, which all bind their ligands to the amino terminal domain, and then the amino terminal domain undergoes some conformational change that is then transmitted down into the transmembrane domain by the amino terminal domain having contacts. Okay, and then here's the C-terminus here. So the major members of this family are then the Frizzled receptors, the ligand for which is a peptide. It's the peptide Wnt. Now, these receptors and the ligand Wnt are extremely important in developmental biology. Okay, so in embryology, these are extremely important signaling pathways. Okay, they also become extremely important signaling pathways in cancer. Cancer cells often use these to aberrantly high levels. Famously, colorectal carcinoma generally has these pathways used at very high levels to promote growth. Okay, but in the healthy adult, these pathways are not major. I mean, they are used in intestinal stem cells, okay, but in most cells of your body, these pathways are not majorly being used, okay, but they are extremely important in development. Right, so again, I'll say that there isn't just one Frizzled receptor. I think there are actually around 10 of the things, okay. Then the TACE2 receptors, again, there's not just one TACE2 receptor, there are many TACE2 receptors. These are really important in gustation, okay? So the gustatory system in taste, basically, they're found in taste receptor cells, and they bind to ligands within food in order to activate pathways that are important in signaling the taste of food. Okay, so TACE2 receptors there. Okay, so the final thing that I should say is that there are 24 Frizzled / TACE2 family members. Okay, now if you do the maths from what I've told you so far and add up the total number of G protein coupled receptors that there should be, i.e. you take 701, which is the number of rhodopsin family members, plus 15, which is the number of secretin family, plus another 15, the number of glutamate, plus 2 times 24 of the adhesion and Frizzled / TACE2, that does not come to over 800. It gets close, but it's not over 800. Okay, so how do I explain this? Well, there are actually 23 members that have not been put into any family at all. Okay, they are in no family. They can't be ascribed to any of these families. They're not sufficiently like any of the other members in these families to be grouped into those families. Okay, right. So that's how you get over the 800 G protein coupled receptors in humans. Right, okay, so we'll call it there for this video, and in the next video what we'll do is we'll firstly move on to the Ballesteros-Weinstein numbering system for G-protein coupled receptors, which is something that if you read the literature you just can't escape from. Okay, so we'll talk about that, and then we'll move on to the diversity of G-protein coupled receptors, so we'll discuss how actually we've discussed this basic structure that they all generally have. But we want to look at how actually within when you look at different G-protein coupled receptors, there is a lot of diversity there. There's diversity, huge diversity in the amino-terminal domain. That's what we've just discussed. Okay, we'll discuss a bit more of that by looking at the example of the sphingosine 1-phosphate type 1 receptor, which has a lid over it, and rhodopsin actually also has a similar lid. Then what we'll do is we'll discuss variation in the transmembrane domains, and then what we'll do is move on to how activation occurs when the ligand binds, what conformational changes underlie the G-protein coupled receptor going from the resting state to being in the activated state.